Hello, and welcome to this EpigenomicsNet interview with Professor Geoffrey Craig. Jeff joins us as part of our focus on the in utero epigenome. Jeff works in the Environmental and Genetic Epidemiology Research Group at the Murdoch Children's Research Institute and is an honorary associate professor within the Department of Paediatrics at the University of Melbourne, both of which are in Melbourne, Australia. His work focuses on epigenetic changes associated with early development and the link between environmental factors for development and disease. Jeff, thank you for joining us.
Thanks, Peter. It's glad to be here.
Absolute pleasure. So if we jump straight into the questions, my first one with you. You were the co-lead author on the recent IVF methylation paper. Could you please give a quick introduction to your rationale for investigating this area?
Well, I work on the role of epigenetics in mediating the effects of early life environment on risk for later disease. And one of the early life environments that has been studied before is IVF, because different forms of IVF are related to taking germ cells out of the body and putting them together and to fertilize and then putting that fertilization product back in the body. These are environments that the early embryo doesn't usually encounter. So we and others had the hypothesis that that's going to change the epigenetics of the embryo and be able to pick that up at birth. And in addition, we use twins to try to get a good idea of the relative effects of genes and environment on the epigenetic differences that we see.
Okay, excellent. And from the findings you found from your investigation, were they surprising to you, or did the results not come as too much of a surprise?
Well, the first results that we found that there were very few marks significantly associated with IVF. So we looked at 54 twin pairs, and we used the genome-wide method, and we only found a very small amount that were significant after adjusting for multiple testing. So that wasn't too much of a surprise. We knew that some studies have found epigenetic legacy and others hadn't. What was a surprise is that 2 of the top 10 genes that we found, differentially methylated regions associated with IVF, had been associated with infertility, both male and female infertility before. So that was the first surprise. And we thought, okay, there's maybe some genetic signal that's influencing genetics. But when we actually looked at, using the twins model to look at the influence, that genetic versus environmental influence, the genes that we found mostly were only affected by genetics a little bit, but more affected by environment, specifically the environment that's specific to one twins, or non-shared environment. So we're still in some way scratching our heads over this, and we tentatively suggest that maybe some genetic factors that influenced fertility may also be influencing the variability of epigenetic marks. It may be making the epigenetics hypervariable, and therefore in twins it looks as though something different is happening to each twin.
Fascinating. And in your study, you utilised whole cord blood cells and cord blood mononuclear cells from newborn twins and employed genome-wide methylated DNA immunoprecipitation coupled with deep sequencing. Why is it you chose to use that method?
Well, we chose that method because we wanted to go beyond the usual microarray method. We wanted to do a better genome-wide approach, something that would give us near genome coverage. So we used the medic sequencing method, and we had good collaborators at BGI, and we know it's got relative. It identifies regions and not specific CPGs, but we thought we'd rather look at as much of the genome as possible.
Okay, excellent. And also, you mentioned earlier, identified no major epigenetic differences in babies conceived by IVF, other than the results that potentially demonstrate evidence for epigenetic modifications that may in part reflect parental subfertility. In sort of future investigations, how will you tackle investigating if these differentially methylated regions observed remain over time, their relation with gene expression, and their sort of potential role within health and disease?
I'd like to work with those who have more details of fertility compared to what we had. We didn't in our original questionnaire. We thought that might be too sensitive to ask, but that would really help to tease apart the fertility versus the IVF. I wrote a review on IVF and methylation previously, and there is that issue of fertility versus the IVF treatment, and also which part of the IVF treatment can affect DNA methylation. And it's amazing that at the time I wasn't aware, but there's some really good animal models of IVF that can really split apart the process stage by stage. So I would I'd ideally work with animal models as well. And also you have a unique subset of individuals who've had fertility treatment and conceived naturally. So that's one variable, the ovarian hyperstimulation. And then those that just have, they're infertile, but they have gift, a gamete into fallopian tube transfer, which means it's as natural as possible inside the body fertilization. So that combination of larger sample sizes, different types of proportions of the population, more information on fertility and animal models, that would really take it forward.
Oh, fantastic. And now for my final question. You identified a few limitations within the study, such as variable cell compositions in the epigenome association scans, methylated DNA immunoprecipitation coupled with sequencing, lacking base pair resolution, a fairly small sample size, and the approach not conclusively determining the cause of the observed IVF-associated methylation changes. How do you plan to overcome this in future investigations?
Well, I think I would try a more higher resolution method, or I would try the ones that I would focus on the ones that the top differentially methylated regions that we identified and study them with a more higher resolution method. We do this in other studies. We identify regions and then validate with a locus-specific method, like sequenom epititer. That's the way I'd go, first of all. I'd also look at the literature to see what others have found, but we did get some relatively strong signals at these top differentially methylated regions. So with limited resources, I'd do that. But otherwise, I would seek collaborators with similar cohorts to make sure that we could cover bases of the different types of individuals and get more information on fertility as well.
Okay, excellent. As I said, that was my final question. So I'd like to thank Jeff for his time, and also to remind our viewers that EpigenomicsNet is on Twitter at EpigenomicsNet, and we'd be absolutely delighted to hear your thoughts on today's interview. Jeff, once again, thank you for joining us.
Okay, thanks. It was a pleasure.
Thank you very much. Thank you.